Hi, this is your first PowerPoint, and this one is over how we can actually set up the scientific method to study humans and human biology. But we're going to start out by looking at a more general sense and just figure out how we study life using the scientific method. To get us started, though, I wanted to go over briefly what to expect with regards to your introductory biology hybrid section. Please note that I'm going to come to your class on the 17th and talk to you about your syllabus, and you will find your syllabus currently in the course content on Blackboard. I want you to pay special attention as you will have an instructor named Judy Glynn who will be actually teaching your lab for you on Tuesdays from 7 to 8:50 p.m. in Science South 106. You can only miss one lab all semester long. If you miss more than one lab, you will actually fail the entire course. So you need to know that right up front. My office hours are actually listed in the syllabus. They are in Science South 115, and my email, tpeters34@stlcc.edu, is the easiest way to reach me and the fastest way that I will get back to you. There's actually two exams in this class. There will be a midterm and a comprehensive final. It is not a self-paced course, which means you actually have projects such as weekly readings, PowerPoints, and also assignments that are due on time to me either by email or by doing something called an inquisitive. I will come to your lab and explain a little bit more. I will hand out a copy of both your syllabus and your unit schedule, explain to you how to read these a little bit more. But if you need any more information, a lot of that's found in the course syllabus, or please email me at your earliest convenience. First thing I want to talk about is what are we define biology as. Ology refers to study of, and bio refers to the study of life. And so when we look at biology, it's really the scientific study of life. And what is life? What is science? On the right side was a question that I posed to a class prior to your lecture. And I indicated to them I wanted them to come up with one word that they would say was, what is science? And you can see on the right side here, this is a word cloud. If a word is much larger, it means more people answered that word. You can see a lot of people had very different ideas of what science would be. Some were very deep in thought and said science is science. But science could be living things like trees or dinosaurs, or the looking at things that are left behind like fossils, looking at things like exploration. We're going to talk a little bit more about what biology and science are. Specifically, we're going to look in this semester at what genetics is. You can see on the right here, we're going to look at what a scientific study really pertains to. There's a series of steps that's involved in looking at any kind of scientific study. The first thing that we have to think about is whenever we want to answer any kind of questions in the natural world, we use the scientific method. It really is looking at what a cause and effect might be. If you do something, then something occurs. But how does that something occur? It also allows you to distinguish between things that are just happening on their own or things that happen because of a specific reason. There are 4 key steps to every scientific study. You must know these. It's a learning objective. These are observation, hypothesis, experiment, and evaluation. You can see the first thing that you might do is ask a question. This is seen on the flow chart on the right side. Your question might be, why is there frost on the window of my car? You might go online on a Google search and see that other people have already looked at what might happen with regards to that background research. These are both thinking about observations. The hypothesis is really an if-then statement that tries to look at if the temperature drops below 32 degrees, then frost will occur on my window. It's really sort of asking a question. You use the experiment then to set up a series of variables, which I'll get into a little bit more here in a few minutes, and you try to understand why does that frost occur. So you might use a closed box and change the temperature or change the amount of water in the container to determine how frost occurs. Then you analyze your results and you go back to your question, your hypothesis, and you see if your results lined up to what your experiment was. And then you determine not whether you have proven your hypothesis, but you see, do you support it? And if you do support it, then the hypothesis is true and you report those hypotheses as a conclusion. If you see that they don't line up to your hypothesis, you might have to do this all over again and go back and restructure a new hypothesis and a new set of experiments. The scientific method is divided into those 4 parts. The first is observation. This is using your senses like vision or hearing or any kind of extension of your senses. It tells us what are interesting things, what are important or confusing. And often we do research to see what's already out there. We have to know what's already out there. It's not important sometimes for you to continue to grade or to put out things that are completely brand new. Sometimes we just want to sort of build on what's already out there. So your observation is your first. The third step is the hypothesis step, and this is really looking at statements, if-then statements, that really give you the best guess to the answer of your problem. These are really predictions of what you want to test in your experimental design. And the experimental design really looks at the third part, which is gathering reliable and repeatable information about what's going to happen based on your hypothesis. These experiments are designed with variables and controls, and they're designed to really look at just one change and the outcome, basically. We generate results in the form of tables, charts, even on graphs, and we try to use this to understand whether the hypothesis is supported by the experimental data. The last step is really evaluating that data and determining whether the hypothesis was supported by the experiment. For instance, do you accept or reject the hypothesis based on the results that you have? If they are results that match up with what you accept as your hypothesis, you accept the hypothesis, and then you might formulate new questions to ask. But you may have to go back to the drawing board if your results don't match up with what you predicted as your hypothesis. So let's look at a key experiment. This was Kettlewell's experiments looking at the turn of the century. And he looked at an observation, basically 2 moths, white moths, dark moths. And if you looked at a natural phenomenon, he saw a huge explosion of the dark moth population over the prior more plentiful light moths. And what you can see on the bottom are different colored tree barks. If you had a dark moth on a dark tree bark, they would actually blend in more. And the white moths would actually stick out more, right? And you can see on the right panel, this is a tree bark that actually has more whiteness in it. So the white moth is definitely going to blend in, whereas the dark moth sticks out here. And basically, he saw that by 1895, 98% of the moths were dark. So he wanted to ask a question. And his question was, if there was a hypothesis, he wanted to know whether the camouflage was protecting these moths from being picked off by birds. Now remember, hypotheses are not proven. They're either accepted or rejected. And Kettlewell basically put a dot of paint on each moth and looked at the polluted and unpolluted areas and then counted the total number of moths to see whether his experiment lined up for this. So his control for the hypothesis were moths that were not actually dotted on their wings. These were just natural moths in the area. And he basically looked at the following data. And I want you to look at the first line. These are clean trees, and he looked at light moths and dark moths that were captured off both of them. And he saw that there was about 13.7% light moths captured off clean trees. And he saw that the dark moths that were recaptured were much lower. Okay? These were trees that were darker, and it was harder for them to capture more of those dark moths because they couldn't find them as well in the bark. Now look at the second line. This is the one that's more interesting. You can see there's not much difference on polluted versus clean trees in the number of light moths that were recaptured. But these polluted trees, which were lighter colored, actually had many more dark moths that were recaptured because these polluted trees were whiter and it was much easier to pick those dark moths off. So this does seem to support the hypothesis that the large fluctuations in populations of moths that could be there are based on camouflage. But what are variables in these experiments that they set up? Well, we're going to talk about 3 different kinds of variables. The first one are called independent variables. I want you to think of these as things that you change or I change, independent variables. Okay? Those are the things you're changing in the experiment. The dependent variables are the ones that are resulting changes based on what you're looking at. So dependent variables are the things you're measuring in the experiment. Control variables are really everything that's kept the same. Okay? So let's use an example here of looking at how music might affect plant growth. And the question that we're going to ask, or our hypothesis, is how does the type of music affect plant growth? And we might look to see that the independent variable in this experiment are the things you're changing. The type of music, it might be rock versus classical music. And the actual height or the length of the plant might be the plant growth that you measure. That's your dependent variable. And your control variables might be that you use the same size plants, the same amount of light, you water them the same, they have the same amount of soil. Those are all control variables in your experiment. The second thing that we have to think about is looking at the sample size. How large of the experiment should you be setting up? So what if I told you that there were only 5 moths captured in Kettlewell's experiments that we looked at today? That's probably not enough for you to be convinced that the results were actually strong enough to make you feel like those are the right results. Okay? So ideally, we really want to have as many as we can to look at in our experiments. The sample size, which is usually denoted by a small n equals, is basically going to look at how many are possible. So typically, a smaller group, and you scale them up in your experiment. So the first set of experiments, you might look at 15 or 20, and then you might look at slightly larger numbers in the second group, and then larger groups after that. So how big of a sample do we need for reliable and repeatable results? This differs based on the experiment, and you really have to look at these each time. One of the important things to think about when we look at sample size is what would you consider to be the right number for an experiment to be valid. So if I flipped a coin and I wanted to determine whether I got results being 50% of the time I got heads, if I only flipped that coin 5 times and recorded the percentage of heads, this might not be as effective or as well supported as doing this 50 times. So increasing the sample size really helps with giving you a better way to either accept or reject your hypothesis. Statistics and science are going to be plentiful this semester. We'll be looking at chi-square analysis a little bit later on in the semester, but they are basically objective ways that allow us to compare results and predictions. It really does get us away from just guesswork and trying to see relationships between two things that might be related. And really, most importantly, that larger sample size gives us more confidence. But we usually have to start with smaller sample sizes and build up to those larger sample sizes. In the next PowerPoint, we'll talk a little bit more about how clinical studies are set up with human studies. Now, are there absolute truths in science? Not really. Absolute truths are really difficult because there's a lot of error in the scientific method pathways. Okay? You can never account for all the control variables. So the key here is when we set up our experiments, we want to have one or maybe just a very few independent variables. We want to have one dependent variable, but those control variables are really hard to control for. Now the sampling and the statistics, not everyone can get every part of the statistics out for every experiment, so this makes it sometimes difficult. Things that might be more colorimetric. Pseudoscience is something that we'll be talking about in this week's lab. A scientific claim that lacks supporting evidence or that does not follow scientific theory. So for instance, if I had a false news type of a situation where somebody came on the news and said that eating lots and lots of fat might lower your risk for gaining weight or being obese, or some new medical procedure. Sometimes this doesn't follow a typical scientific process of asking a question, setting up an experiment, and getting the results, but it's kind of hard to figure out sometimes what pseudoscience is. We're going to spend time evaluating this a little bit more. We also have to think about some definitions. Facts or scientific facts are repeatable observations. Laws or scientific laws are where we see no known exceptions for that particular circumstance. Now the ones in red here are very important. Scientific theory are testable and relatable, and they're findings that are strongly supported, such as the theory of evolution or the cell theory that we'll be talking about. These are strongly suggested to be ways to look at how things work. And we'll be spending a whole unit looking at the theory of evolution, Unit 4. Hypotheses, as I've indicated before, are educated guesses, or they allow us to make sort of an educated guess or a provisional guess at what's happening. And models are a way for scientists to sort of think about how things might actually work in a different way.